I want to thank Scott for the invitation to speak at this fun course, and he's given me a challenge of talking about a large group of therapeutic agents, of which we are all now highly familiar with, and to kind of look forward. So I thought of doing this many different ways, and there's a lot of exciting things going on in bioengineering which are still at preclinical levels. And I thought I would limit my discussions today to those advances, either in the current use of these agents or advances that are currently in the clinic. These are my disclosures. I thought I would do this in several stanzas, kind of give a 30,000-foot view of why TNF is still an enduring target of interest. We'll talk a little bit about the first-generation drugs. Then we'll talk about what we know about TNF failures, because this is really what it's all about. And then finally we'll talk about new agents, both some small molecules, a little bit, some novel agents, and I'll close by talking about the migrainous area of biosimilars. A few questions to ponder. We now have 5 approved TNF inhibitors. Are they really individual drugs, or are they a class? Does failure to respond to a TNF mean that you're a class failure? And do we understand the nature of these types of failures? And do switching agents mean anything to us? Are these agents equivalent in efficacy and safety and this futuristic compound? So, it's always important to look at the history of our field. And the history of TNF dates back to over a century. William Coley at the New York Hospital was the first to identify the potency of a substance in the blood that had the ability to attack tumors, but he did it in a rather novel way. He infected people with bacteria, first live bacteria, then killed bacteria, and made some intriguing observations that this produced some type of host response that in some patients resulted in necrosis and shrinkage of tumors. It was really almost 100 years later when Old in that same city demonstrated that in mice that were sensitized to Mycobacterium and then challenged with LPS, they could make an endogenous, a host-derived product that could lyse tumors, as shown down on the lower left. And I think is this the there we go, great, lower left here, showing tumor necrosis. It took a few years, but this was sequenced and cloned, and then attempts were made to produce antibody to this, and clinical developments began in the late 80s and 90s. And by the end of the 90s, actually 1997, 1998, we had our first approved drug. And since that time, this is really inaccurate, this was 2009, it's probably over 3 million people globally have been treated with TNF inhibitors. So this is an incredible database of to which to draw observations on efficacy, toxicity, pathophysiology. But we're here at the leading edge of this now, and we need to know how we can improve their use. The availability of a class of drugs that inhibit a central inflammatory pathway have provided some of the grist for the notion of this immune-mediated inflammatory diseases. And whether you buy into this or you don't buy into this, it has been functionally useful. That there are diseases that share certain pathways that can be treated with similar agents, even though idiopathogenically they may be remarkably different and their clinical expression may be markedly different. We have this monotonous ramp up for the beginning of all of these autoimmune diseases, where we think that there's a genetic background combined with environmental factors and accelerants that lead to perturbations of the integrated immune response. Many of these perturbations result in the elaboration of a host of inflammatory cytokines, of which TNF is merely one, a very early and critical one, albeit, that participate in the damage seen in these diseases where these drugs are approved. Thing from Crohn's disease to rheumatoid arthritis to JIA to the spondyloarthropathies and psoriasis and other indications that may be coming. But the real horizon is the fact that these same molecular markers are participating to a certain degree in a wide variety of other conditions, ranging from infections such as HIV to aspects of aging such as cardiovascular disease, etc. So we have taken some lessons from this and are moving forward. So, you know, the biology of TNF is now well known in certain regards. It is produced by a wide variety of immune and non-immune mediated cells. It's expressed as a cell surface as a membrane protein, but it is cleaved into a homotrimeric form that then can seek out its cognate receptors, which are also found on a wide variety of cells. The end result of the ligation of the TNF receptor with either transmembrane or soluble TNF is to transduce a signal which results in either a promotion of inflammation, largely through NF-kappa B, or alternatively to trigger a cell death pathway. What determines which direction that this may go, or what the predominant effect, has to do with the level of stress of the cell and the milieu at which this reaction is taking place. So this gives a lot of opportunities here to intervene. We're going to be talking about events at the cell surface, and Dr. O'Shea is going to be talking about events within the cell in the trans-signaling pathway. This is the family of drugs that we have, and my friend Gary Firestein says that rheumatologists live in a world of earthly delights because we just have all of these drugs available to us. We have all the toys. And these are, three of them are antibodies. They vary in terms of their bioengineering. We still have a highly widely used chimer and infliximab. We have two human antibodies made by different bioengineering models. We have a receptor construct, which by definition does not have an FAB portion, and that has probably something to do with immunogenicity. And then we have a novel compound, certolizumab pegol, which is devoid of the FC portion. All of these share very high degrees of specificity, very high degrees of avidity for TNF, and they all are somewhat equal in kind of a Venn diagram kind of way in many of the diseases, such as rheumatoid arthritis, but then diverge in their efficacy in other areas, such as inflammatory bowel disease. And questions of why this occurs is really not what we're going to be talking about today. I'd like to first mention and just kind of ground us that, you know, the next generation of TNF inhibitors, the current generation of TNF inhibitors, have built upon the groundbreaking work and observations of Cesar Milstein and George Kohler, who received the Nobel Prize for their work in the production of monoclonals in 1984. Started very humbly with a letter to the editor in Science in 1975, where they had described their production of a monoclonal protein in a purely murine system. They were British Research Council researchers, and the story is that they had sent this observation on to the funders and said that they were very excited about this, and they were going to make a manuscript, and they wanted to work with them, and they it is said that Caesar Milstein kept a letter that he got in return on his laboratory till he died. That they said that, well, this is kind of interesting, but we see no commercial application of this, and they never patented it, and so such is life. The rapid moving forward of the development of TNF inhibitors has moved from the murine system, which obviously was incompatible with human therapeutics, to a chimeric system where hypervariable regions were annealed into a human framework, leaving a molecule that still had xenogenic epitopes, which could elicit an anti-drug response, but were largely human in the rest of their construct. Over time, these antibody combining regions were reduced to keep only the complementarity determining regions, further limiting xenogenic components of these antibodies, but not eliminating them. Finally, with advances in bioengineering using both ex vivo phage expression systems and now systems such as the proprietary Humax mouse, where a mouse has had its endogenous immunoglobulin gene silenced and human immunoglobulin genes inserted into it. These mice can be challenged and produce antibodies via a physiologic maturation process in secondary lymphoid organs that has not been possible before. There are now advances going beyond this with aglycosylated proteins produced in non-mammalian cells. We'll have to wait as these move into the clinic. But that is kind of something to keep in mind when we talk about immunogenicity and what we're doing. So these agents, again, as we look at them here, they are different in many things. They're different in the way that we use them. They range from sub-Q as infrequently as once a month with golimumab to those which can be given as frequently as twice a week. There's a single IV preparation at the present time that can be given every couple months. They have different PK characteristics, and they have different degrees of immunogenicity, and I'll talk about this in a little bit of time. You are all aware that this approval, both by EMEA and FDA for these, they are not all the same. Part of this is that some of the newer drugs have not been put into full clinical trials in all diseases. But drugs such as etanercept are clearly not effective in diseases such as inflammatory bowel disease, and are not very effective in the non-approved indications of granulomatous infections, but they're, in contrast to the antibodies, in particular, most of the data coming from infliximab, which shows that they are more effective. So this has created a kind of a list of questions that have yet to be answered as to why this is. Is it differences in PK, PD, FC-associated responses, basic immune disruptive responses? We won't be able to answer that today, but I'm trying to make the point that there are differences. The leading clinical question today in the use of these drugs is that, well, we know that they are highly effective for all of these diseases, but A, why aren't they effective for everybody with these diseases? And B, when they are effective, why do they fail? and we have a taxonomy of this, and I'll show you a little bit about this. I'm going to talk to you about a little bit of definitions. I'll talk to you about where we're at in genomics. I'll make a few comments about therapeutic drug monitoring, and then talk about immunogenicity and switching. So one way of looking at these drugs in terms of how effective they are, and you can look at this as a therapeutic index here, and going down means that things are working good. A drug that is highly effective shuts down disease activity and stays down. Occasionally, in rheumatoid arthritis, and not too dissimilarly in inflammatory bowel disease, some goodly percentage, whether it be a quarter or a third or more, just don't make a rich response right from the get-go, naive to this class of drugs. That is less common than this pattern. And these are people who initially respond, but then over time, it goes away. So this would be a primary failure. These people would be varying degrees of partial secondary failure. And this person down here, who actually was doing pretty good, this drug was stopped because there were side effects. So we understand the side effects pretty clearly. But why these groups are occurring right now is not well known. So the variability of response could be differences in PK, PD, based on different immunogenic mechanisms of action. They could be based on host factors. Not all diseases, and we heard so elegantly this morning talking about multiple sclerosis, different phases, different compartments, and I suspect that our diseases are very similar to that as well. Initiation phases and propagation phases, scarring, etc., all may contribute to this. So what do we know a little bit about this? Well, everyone who has made a TNF inhibitor has tried valiantly to come up with a signature, whether it is using biomarkers or more recently to find a genomic signature to say, can you tell me who's going to respond to this drug and who's not going to respond to this drug? I can't even imagine the millions of dollars that have been put into these. And to date, there is only the puniest of evidence that we have tools. And not that there are not genomic influences, but in terms of a Bayesian model of clinical decision making, these are nowhere. They give you a little bit of a whiff, but it does not occur. Now there are some very robust studies that are in the early phases. This is called Batter Up. This is using a very robust clinical and genomic database out of the Brigham, and looking at this in as good a detail as possible. This is being funded by multiple sponsors, primarily by Biogen, and we're far away from this. And this will be matched to good clinical responses, toxicity, etc. But that is the short and the long of it. These references will guide you if you want to read more about it. Now, more interestingly and more practically, to a certain degree, is the notion of therapeutic drug monitoring. Now, this is a common thing. We do this in a lot of diseases. We look at calcineurin antagonists, and we look at antiarrhythmics, and we look at anti-seizure medications. Is there the potential to therapeutically drug monitor this family of biologics? And that is actually getting down to the issue of measuring drug levels. It now appears that there's growing data that there is a good correlation between drug levels in general and therapeutic responses. And whether it's Crohn's disease or rheumatoid arthritis, if you take the people who measuring at trough, that means before their next injection, and they are non-detectable, you don't have any more drug left. Every study has been internally consistent that those people are having the least in terms of their therapeutic response. Now the question is, can they be used in a clinically predictive way? And a follow-up question is, why does that happen? So the good reasons that would come to mind immediately are, and we heard a little bit about this with Dr. Harold this morning, is that you can make antibodies to these antibodies, and we'll talk about that in a second. That's a easy to understand reason. But all of these studies that have looked at this with appropriately specific techniques show it's really only a partial explanation, and that there is still a significant amount of people who have non-detectable drug levels at trough who don't have antibodies. So what does that mean? So the other thought is that, well, many of these studies have also shown a thread that it's patients with the worst disease that seem to do this. So perhaps they have the highest degree of inflammation, and maybe these antibodies are being sopped up and cleared by physiologic clearing mechanisms. It's a hypothesis that is yet to be tested. Lastly, we know that we all have genotypic variants in our FC receptors and our ability to clear antibodies, particularly in bound form. And are there genotypic features that have correlated with this? These are all in progress. There's been some recent data, I didn't make a slide for this, there have been SNPs in immunoregulatory regions, particularly such as in IL-10 region, that have correlated with presence or absence of antibodies. There are several studies that I'm going to kind of rapidly move through. This is one of the studies that you can look at in terms of inflammatory bowel disease, showing a rich correlation between drug levels and higher rates of remission and endoscopic improvement, but no correlation with anti-infliximab levels. Immunogenicity, clearly a big part of this. How much of a part of this? It has been shown that people with the highest antibodies will have, when I say antibodies, I mean antibodies to drug, will have the lowest level of drug at trough. Secondly, it's been shown that this has clinical consequences, as I've just told you, plus correlating with toxicity, particularly local injection reactions, infusion reactions, etc. Right now, while you can go out to your favorite send-out and get an assay done, these are technically difficult, and all of them are plagued with certain degrees of limitation. They're very hard to measure while you still have drug in your system. In rheumatic disease, they're plagued by interference with rheumatoid factor. They're not standardized, and I'll show you how different that can be in a minute. The mechanisms of immunogenicity can be quite different, and we were talking after Dr. Harold's talk about what could be responsible for this. Well, it's easiest to understand when you have a chimer and you have more xenogenic determinants to react to. But even in the absence of chimeric antibodies, when you have human or humanized, you still have small degrees of bioengineered regions which are not found in the germline, so-called anti-idiotypes, which can react to. We also, a lot of these antibodies are IgG subclass 1, and those will contain allotypic determinants, GM, and they're all kappa, and so KM determinants, and these have been demonstrated to be the rich source of antibodies in certain patients. Finally, they can form immune complexes, and that can be the source. The summary of all of these data, I can tell you from the multiple groups that have done the best work with this, is that these antibodies are not uncommon. Most of them react with FAB portion, not FC, most of them. Thirdly, while all isotypes are represented in these anti-drug antibodies, the predominant isotype is IgG. Interestingly and inexplicably, in several studies, about a third of the antibodies that are produced are IgG subclass 4, and that seems to be telling us something, but we don't know what. And that lastly, most of these antibodies can form immune complexes with up to 3 antibodies per therapeutic molecule. So those are some summaries found in some of the references I've given you. The clinical correlates of this are decreased function and increased toxicity. The most interesting paper to come out on this is very new, just in the last months, by the Dutch group. And this is a several-year study of rheumatoids who then post hoc were looked at for presence of antibody to the therapeutic agent adalimumab, a human monoclonal antibody. Now, if you look at the package insert for adalimumab, it says that antibodies are found in about 5%. And using their assay, that is exactly what was found. In this assay, if you look at the percentage of antibodies developing over time, as you can see, over about a third of these people will ultimately develop them. And these were done by liquid phase RIA, which is probably the standard to do this and gets rid of interfering rheumatoid factor and other things. So this is really just looking at the mean here. There are patients with low, medium, and high, and I just want to show you a couple graphs that tell us the difference. Forget the colors because actually the colors came out good on here. Look at the colors. These are people without antibody. These are people with medium, and these are people with low. And these are looking at clinical responses. And as you can see, there's an intense correlation. Furthermore, if you look at these are arthritis. These are arthritis patients. So the higher the response, the higher the curve goes up, the better the arthritis response. Whether it's sustained minimal disease at these levels, if you're not rheumatologist, they don't mean anything to you. But no antibody, medium, and high antibody were very, very, very consistent throughout this study. So this taught us quite a bit that this human antibody can be intensely correlated with these levels. And this has suggested to certain people that this could be clinically useful. The final thing that you should know about this is that it has been known for over a decade that concomitant immunosuppressives, even low doses of methotrexate, can attenuate the anti-drug antibody response. So these things need to be considered. We were just talking at lunch about a new biologic, pegloticase, used in gout, highly immunogenic, no strategies for it at the present time. So this is what I will close the stanza looking at what I think is a new PK taxonomy to the use of biologics. So I think that secondary failures now are people who have had some response, who are not responding, who either have non-detectable trough levels or very high levels of anti-drug antibodies. And those people, it has been demonstrated, if you switch the TNF agent, etanercept to adalimumab, infliximab to certolizumab, any way you switch it, they still have a very good chance of responding because most of these antibodies are to these FAB determinants that we talked about. On the other hand, if you are failing this drug at any point in time and you still have drug in your system and no antibodies, these people are probably pathophysiologically failing this drug. And those are in rheumatoid where you have tons of options, and we can target their B cells or T cells or IL-6 or other routes. And while the assays to tell us this are not quite ready for prime time, I think they're moving in that direction. So that's kind of interesting. An area that I quickly will touch upon is the potential for small molecules to inhibit TNF. And we have, none of these are approved, and I'm not going to talk about JAK inhibitors or SYK inhibitors. I will rather just mention a few more specific areas. I'm not going to talk about TACE. TACE is the TNF cleaving enzyme. This is not ready for prime time. This has failed in early clinical trials. There are several immunomodulators, particularly of the thalidomide and thalidomide congener family, and phosphodiesterase inhibitors that deserve at least a little bit of talk. So thalidomide, a drug you're all familiar with, it now has several congeners which are designed to be less teratogenic. We thought that we understood thalidomide at one point in time. We really don't understand thalidomide at all. Thalidomide, there have been nice studies done in IBD showing that falling disease activity and falling TNF. On the other hand, for over a decade, we knew that thalidomide is also a very good drug at healing apthae and esophageal apthae, yet TNF levels go up when you give it, and it's still very effective. So, as I was saying at lunch, I said, the field of immunology is the field of surprises that prepares you for future surprises, so be proud of it. The area of phosphodiesterase inhibitors is a complex area. Multiple isoforms of this enzyme. It's ubiquitously distributed. The PDE4 is found predominantly in hemopoietic cell lines, and by inhibiting this and with its secondary rise of intracellular cyclic AMP, multiple cytokines can be affected. So these are not selective TNF inhibitors, just like thalidomide and lenalidomide and congeners were not selective inhibitors. They're really immunomodulators. But this notion has been around for a long time, and there have been drugs that have been used to do this. But unfortunately, all these drugs have caused terrible side effects. Either put you to sleep or you can't stop throwing up. So they were going nowhere in a hurry. There's a new drug that is in clinical development called apremilast, which does this similarly, but has a better toxicity profile. This has been looked at in small numbers of patients with inflammatory arthritis. It seems to be modestly well tolerated. This is the drug run-in phase. Placebos who then got drug again. This has been associated with falling cytokines. Whether it's going to make it or not, I don't know. Lastly, let's talk a little bit about the novel biologics. And there are some interesting advances in bioengineering that are making their ways to the clinic, and we'll just make a few points about these. Dominant negative constructs are not in clinical trials that I know of, but this is such an interesting notion that I think it's important. So the idea of a dominant negative is that, so here is transmembrane TNF over here, and this is the TNF receptor, and when this membrane protein anneals to its cognate ligand, you'll get triggering of this cell. And whether we need to talk about reverse signaling or whatever else is going on this way and this way, that is an important immunologic homeostatic mechanism. And there have been elegant preclinical models to show that if you leave transmembrane signaling intact, you can protect the host from a lot of stressors like listeria challenge, etc. When you give a drug like etanercept, you can bind to the membrane and neutralize this. Any of these drugs will do this to a certain degree. This may be the weakest. On the other hand, TNF also is cleaved, and it's in its soluble form, and it binds to its receptors and does the same thing. A dominant negative is to create one of the trimeric components of the homotrimeric TNF that is functionally deficient. And these are in apparently a rather free equilibrium. So if you give a drug that is composed of these dysfunctional trimeric units, they will interchange with the soluble TNF, forming a dysfunctional overall TNF molecule. It does not interchange with the transmembrane portion. So there have been a number of studies now done in murine systems where you get very good TNF inhibition, but you maintain transmembrane signaling. So you can shut off inflammation but still challenge these animals with listeria or other pathogens, and they respond to it. It's a very fascinating and very interesting notion. I've given you a few references here if you would like to follow up on that. The real horizon that in bioengineering right now is and I don't want to sound old-fashioned about this. There are some great ideas out there about how to improve on antibodies. On the other hand, you know, these few billion years of getting to these antibodies has kind of told us something. So when you make say you're making a better antibody, a bio better, you better have a lot of evidence for it. So there are no peptibodies in clinical trial, and those for TNF, yet there are for many other diseases, and those are just random peptides that have ligand specificity and can be either pegylated or Fc'd. But there's this smaller antibody. These are called DMAbs. So we know that if you can just take the variable region of the light or heavy chain alone, it can bind its cognate antigen in nanomolar concentration. So these are bioactively small peptides. They have very short half-lives, and they're not very useful because they would just be eaten up. But you can make them endure. And so one of the technologies is to take these, and I'll show you a picture of this variable light chain, and then put it on a truncated Fc where you have a little bit of hinge, so it could probably bind to a couple ligands, but you've actually attenuated its complement binding activities, and you can bioengineer this any way you want. It is very highly avid. Surface transposon studies show this. It has a respectable half-life. There's a thought that if it's this much smaller, it could have better tissue penetration. So, as they say, equal efficacy at half the size. So we have to see if that means twice the benefit or half the benefit or whatever. But these are now moving in through phase 1 for a number of diseases, including antibodies molecularly targeting TNF. I've given you the reference for that. I thought this morning Dr. Harold's talk quickly mentioned that their lead molecule, their anti-CD3, was bioengineered, so it did not bind Fc receptors. And I think that this is the kind of thing that we are seeing. This post-translational glycosylation is extremely important in antibody engineering. Up to 30% molecular weight of an antibody can be glycoproteins, and these can be modified in varying ways, site-specific, globally, to do a lot of different things. You can certainly make them not bind to Fc receptors. You can enhance their binding to Fc receptors. You can alter their PK. This is now being done. There's a candidate molecule. Xenocor is a company that is making their entire raison d'etre on post-translational glycosylation of monoclonals. And they have an array of things where they can take these antibodies and then make them selectively bind to inhibitory receptors like FcR2b, so combining a double punch. So I look for these as next wave improvements, but they're not really ready for prime time. All right, so finally, biosimilars. This is such a great topic, and I'm probably not the right guy to talk about this. And there's only a couple of people that can actually talk about this, because you have to be a scientist and a lawyer. So I'm somewhere in between. I've seen this slide before. I think this is yours, Scott. I credit to Dr. Plevy. But it makes a nice point that, there's reasons why people are trying to make biosimilars, because this is a long road And even though, it's getting shorter and there are new discovery processes and it's becoming a little more high throughput, to get from the idea to a molecule to a clinical trial, and then get it out there with its 12-year patent, it costs hundreds and hundreds and hundreds of millions of dollars, and we've seen drugs go down at the last minute. So there's a notion that we would like to do something. Also, this is a very lucrative area. It's believed that TNF inhibitors are already over $12 billion a year, and that biosimilars have the potential to erode this. So as you can see, the people who make this is serious business for them. So, whether these drugs actually occur is not. So what are we talking about here? First of all, I will use the term biosimilar. And there are other terms that have been used in the literature, follow-on biologics, etc. But biosimilars are drugs that have similar structure, similar physical, chemical, and immunologic properties that have been demonstrated to have similar efficacy, similar toxicity, and interchangeability. That is what a biosimilar would be. And there is legal language within the healthcare reform bill that documents this and provides quite a bit of financial incentive to develop biosimilars. Why? Because this is costing all of us tons of money. We saw the $34,000 a year for belimumab. That's half the price of pegloticase. And so if we could make a cheaper biosimilar, that would cut down on our overall healthcare costs. The problem at the present time in our country is that this is a law. Now, this law is then given to the FDA, and they are to create a roadmap, a guidance document of how you do this. And they haven't done this yet. And there's reasons for this. I don't think it's for lack of will. This is just an extremely complicated process. And at the present time, there are companies that are moving very rapidly, but their products are being dealt with on a one-to-one individual basis. So the idea is that we have biosimilars of small molecules. We call them generics. But biologics are not just a monolithic compound. There are biosimilars already for certain growth hormones and drug agents such as insulin that are made through bioengineering. And these have been pretty effective, although there have been speed bumps along the way. These are not immunoglobulins, and these are relatively simple molecules compared to this. And I'm going to come back to talk about the problems of creating a biosimilar to 150 kilodalton complex multiple chain antibody. These are drugs where there are already biosimilars for biologics, but as you can see, as I say, they all fall into the growth factor hormone area. The challenge of making an antibody that is biosimilar to another that met all my criteria is that these are all physical and chemical properties of immunoglobulins that have to be matched up. And they have to do with their binding, their effector function. This is found both within the variable and the constant region. Even if you have the same amino acid sequence, their post-glycosylation patterns and their quaternary structures may differ. So the question is this is a tall tale. We don't have anything available. And you should ask yourself now, what biosimilars are you aware of? And, if you answer that question and you are not aware of these biosimilars, and this is just in TNF, there are a lot of biosimilars. So there's biosimilar etanercept being used in China, and there is a biosimilar called Etanar that is being used in Colombia. This is the only drug that I've seen actual clinical data for. There is a biosimilar in development for etanercept fusion protein in Israel right now. There is a second, a Taiwanese TNF inhibitor, Tunex, which is already being rolled out. And then finally, there's this life science kind of infliximab knockoff being used at the present time. So the issue for us to think about is that, as I say, if you knew that, and there's been some industrial intrigue with these of how the molecular structures were acquired, how the cell lines were acquired. So, I mean, as I say, it could be like a John Grisham novel right now of what's going on. But, how will you prove bioequivalency? If I have a drug, Tunex, and I show that it's equal in a group of rheumatoids, will I be given the indication in JIA, psoriasis, and psoriatic arthritis? How will I prove safety? Safety is found in regulatory registration trials. These are going to be very small trials. And lastly, how will you demonstrate interchangeability, particularly from the perspective of immunogenicity? And it means that you can be on Enbrel, and you stop it and put it on Tunex, and then go back to Enbrel and go back to Tunex. Is that all safe? And how will you design a clinical trial to do that? What will you be offering a patient who participates in such a trial? You're offering him nothing in terms of efficacy. You're only offering him the chance to find toxicity. I don't even know how an IRB is going to do this. So I'm not like a skeptic. I have a headache every time I talk about this. Finding the molecular sequence is easy. Taking this from now through regulatory will be a continuing issue. So those are my thoughts. Thank you.
There might be time for a couple quick questions, and I'm happy to step back.
Cytokine receptor antagonism probably is another strategy for blocking the cytokine-mediated inflammation. So, do you know if there are any anti-TNF receptor agent in developing? Or a second question is, as for the TNF, there's two TNF receptor 1 and receptor 2. They are mediated. The bioactivity is kind of different. I think probably the TNF, the side effect of TNF antagonist is because of its TNF totally blocked by the antibodies or FC. Do you think it's a good idea to develop selective TNF receptor antibody?
First question, I know no drug in human trials that targets a receptor per se. Second is, R1 versus R2 as a construct. They're both highly avid. There's this notion of receptor passing. It has not been exploited. There was an earlier drug that used the type 2 receptor that was effective in its first iteration, but as it was bioengineered, it lost activity. So I don't know where that would be going.
Thank you.
I have a couple of questions. One is the TNF inhibitors. Even you use the TNF alpha inhibitors, the still few percent or the almost 20 or 30% people doesn't affect these drugs. So how to control the 20 or 30% of the peoples?
Well, those would be what we would now call primary failures.
Yeah, right.
And, you know,
That's right.
those probably have some different pathophysiology going on.
So you need a next step of the
You need other target for those people.
yeah, because we are doing the anti-IL-6 receptor antibody. That is very effective than the TNF alphas. Emery says the TNF alpha failure using the anti-IL-6 receptor antibody. That is effective.
That would be one strategy. Yes.
Yes, right.
Thank you.
And another one is the next generation of TNF alpha. You use there are another antibodies, small molecule or the large molecules. How about the small compound, almost a few hundred molars, few compound which bind the TNF alpha or the receptors? These are inhibited TNF alpha or not? You generated such small molecules inhibitors?
Like peptibody technology. Yeah, it's moving forward, but not in humans yet.
But you know some information about the such small molecules inhibitors of the TNF alpha. Some of the company developed such small molecule inhibitors.
Most of the time when we're talking small molecules, we're talking intracellular signaling. Yes. Dr. O'Shea is going to talk about that right after us.
Okay.
Thank you.
Go ahead.
Right.
A couple more quick ones.
Regarding the appearance of anti-antibodies, maybe I zoned out and missed it. Did you talk about whether patients were on methotrexate or are there other agents?
One of the references, and I probably didn't make this clear enough, yes, I think that's a vital component and one strategy to decrease immunogenicity. And the surprising thing to everybody was that even 7.5 milligrams of methotrexate was enough to decrease immunogenicity. Obviously, periodicity of the infusions and all the other things we know about that as well, but good point.
What about in the 2011 anti-adalimumab antibody study you showed? Were those patients on methotrexate?
In the paper, it made statements, but didn't show it in general, that people who were on a second agent, and it said methotrexate or other immunosuppressives, had less, but it didn't show what it is, but it was clearly, and I think significantly less.
Just two questions about biosimilars. The one is in terms of like what cost, what are they actually coming out at? And the second is how are they getting licensed in those countries where they're made? I mean, are they being used?
Those are easy. We don't know what the costs will be. We would love to see this, you gave me a TNF inhibitor that I really thought was biosimilar, and you said it was half as much as any other ones. It would be my immediate favorite. And secondly, we don't know all the regulatory details of where these are being done in these countries. As I say, for all of those molecules, if you PubMed, you do everything you can, you will find only published clinical trial data with one. All right, so this has obviously been submitted to whatever regulatory is on, but it's not public domain.